So now we're going to plot the same plot as we did for UMIs, but using the reads basically. And again, we use top 500 genes. So what we see here is actually much more homogeneous than the UMI plot. So for comparison, if you look, this was the UMI plot where we saw the bad batch quite nicely. With the reads, it's not the case anymore. So it's quite different. And as I said, UMIs are usually much cleaner. Now, if we look at it using the quality controlled data set, it becomes even more homogeneous, which is actually good because we removed different cell outliers, and that made the image more homogeneous. So this is a t-SNE plot of the reads, and again, it resolves the bad batch somehow, but the rest is has a very strange shape, I would say. And now if we plot t-SNE for the quality controlled data set of the reads, there is no bad batch, which is good, and the structure is again quite homogeneous. So you can change the perplexity. This is perplexity 10. This is perplexity 200. And I'm not sure whether it's clear what the difference is and whether you see any useful structure in your data set. So I think that's it for the visualization of the data. Do you have any questions? There are actually other features, other methods in Skater, for example, diffusion maps. We only show PCA and t-SNE, but you can explore the package, and there are more methods to visualize your data set. So the next chapter, I think, is about confounding factors. And Davis is going to continue on this.
Okay, we'll get there in the end. So the next thing for us to discuss is this issue of confounding factors. Everything that we've done so far is focused on sort of figuring out basically which cells are likely to be problematic and trying to remove them from the analysis. So ones that have failed for various reasons we want to identify and remove. There's another big source of variability in data that we'll get, so generally speaking, in genomics, and certainly specifically in single-cell RNA-seq. And this is to do with what we often in the field refer to as batch effects. So it's been very well known for a long time now, especially when we do gene expression measurements, when we assay gene expression, the measurements we get vary a lot just based on where and when the measurement was done. And so this was true of microarrays, it's true of bulk RNA-seq, and it's really also true of single-cell RNA-seq. And sort of the concern here is that we have variability introduced into our data just based on the plate that the cells were processed on, or the day that they were processed, who did the sort, if their cells are sorted, a particular reagent kit was used for one set of plates and not for another. They were sequenced on different days, although the sequencing itself seems to be not a major source of variability. But so as well as sort of the individual variation between cells, say within a batch, we also tend to see large-scale effects between batches. And that's the sort of thing that we want to be able to identify Ideally, we know about it beforehand, before we do any analysis, but we want to also look at our data to check whether these sorts of technical effects or artifacts are present in the data. And once we've done that, we can think about strategies that we could take to ameliorate their effects. So either hopefully remove that unwanted variation from our data so that we have a more focused sort of expression data set that's really picking out the biological signal that we're interested in rather than technical differences between cells. And so that broad picture is what we're going to discuss in this next section. So we'll continue to work with the Bleachak data as we have been doing so we can load that up here as we've done before, identify the full data set, the QC data set, and those endogenous genes that we've used previously. And so again, we can look at correlation, or what we will do is look at a PCA plot again. So as we've done before, do a PCA using the count data, coloring the picture by batch and showing the size of the features. So we can see that probably already apparent from this plot that there's a tendency for cells with a large number of detected features to be towards the right of this plot, and cells with a smaller number of detected features to be towards the left. And something that we can do with the plot QC function in SCADA is to identify which variables correlate with principal components of the expression data. And so if we apply that here, so with the plot QC function, using just the endogenous genes, the type of the plot is find PCs, and we're going to check for the correlation with the variable total features, and using counts as expression values here. Then we generate this plot, and so what this does is it shows the correlation between total features, so the number of detected genes for each cell, and the 6 principal components that are most correlated with total features. So in this particular case, we can see that principal component 1, is the most highly correlated principal component with total features, and the R squared of that relationship is 0.85. So there's a very strong relationship between principal component 1 and total features. And so that's what we saw, loosely in the PCA plot, was that we could see it visually that there was a tendency for principal component 1 to distinguish between cells with different numbers of total features detected. And so by default, this plot then shows the 5 components that are next most correlated, and we can see that, so the second most correlated one is component 5, which is basically uncorrelated, and so it's really this first one, and these plots, the top left one is typically most informative, what's the most correlated principal component with the variable of interest, and here it's PC1. Another thing we can do to explore the relationship or sort of explanatory power of particular variables is to produce another type of QC plot, which I'll describe below. And what we're going to do before we get to the plot is compute the marginal R squared for each variable when we fit a linear model regressing expression values for each gene against just that variable. So the idea is that for each gene, so to take the total features example, for each gene, we'll fit a linear model with Y as the expression values for that gene, and the only predictor in the model will be total features. And we can obtain an R squared value for that model fit, which explains, is the proportion of variability in the expression values for that gene that is explained by, in this example, total features, and that particular variable of interest. And so we can do that for all of the genes that we have in our data set, and so that will give us and get the R squared value for each of them. So that gives us a long list of R squared values across all genes for a particular variable of interest. And so that gives us a distribution of R squared values. And what this plot shows then is the density of that distribution of R squared values for each of these different variables of interest when we fit that linear model with only that particular variable as a predictor. And so what this picture shows, when you stare at it for long enough, so is on the x-axis, this is the proportion, the percentage of variance explained on a log 10 scale. So that's the R squared from the models that we're fitting. And for each particular variable of interest, so total counts, total features, batch, and so on, we have this density curve showing the distribution of the R squared values, those marginal R squared values. And so if we have density curves that are shifted to the right, so with a peak, say, towards the 100% end of this plot, then that's telling us that for a large proportion of the genes in our data set, this particular variable explains a large proportion of the variation for that gene. So, for example, total counts and total features here, this plot is showing us, so the orange and blue curves in the picture, this picture is showing us that for many genes, those variables explain a substantial proportion of the variability. When we look at batch, we can see that's sort of the next in line. So these on the right-hand side, these are ordered by the median R squared. So that immediately tells you which have a tendency to explain more or less variance among the variables that we're looking at. And so you can see for batch, so this quite narrow distribution for batch is really saying that for most genes, batch explains between 1 and 10% of the variation for all genes. And then we can see, go down the list, and for example, of these ones, the one with the least explanatory power for many genes is the percentage of counts coming from mitochondrial genes, and so that mode is almost perfectly on top of 1% there. So for about half the genes, the percentage of counts coming from mitochondrial genes explains more than 1% of the variance, and for the rest it explains less than 1% of the variance. So this one plot takes a bit of sort of getting your head around, but once you're happy with how this works, then we can look at this and say, okay, these distributions that are shifted to the right have a tendency to explain more variability among genes in our data set, so total counts, total features, batch, percentage counts from ERCCs in that category, the particular individual that the cell came from, and the percentage counts coming from mitochondrial genes tend not to explain as much variability in the data set. So this can be useful for getting a first look at particularly explanatory variables that might be worth a deeper look because they seem to be a substantial source of variability in the data set. So we might even run this sort of plot first and then say, okay, if we look at total counts, total features, and batch, then go back to a plot like this and ask, okay, if we use them as the explanatory variables, what principal components are those variables correlated with, and what sort of proportion of the variability do they explain? Any questions about that before we move on? Okay. So in this particular case, we know about the batch because it was clearly, it was part of the experimental design. They sought to investigate batch. There are also sort of processes, technical and biological, underlying the data that can lead to variation between cells that we might want to compensate for. And sometimes these are not always known a priori. Like we know which cell was processed on which plate and so on, so that's a known batch effect. But there are other processes that might be active, like cell cycle phase, for example, that is a source of variation in the data that may not be what we're interested in studying. So it's a nuisance sort of source of variability there. But we don't know a priori which, unless we've measured it some other way, which cell is in which phase of the cell cycle. And so that's where this method, SC-LVM, was developed. It was developed for that purpose, to try and use annotated cell cycle genes and their expression levels to infer, essentially to infer the cell cycle phase for each cell, and then to remove that source of variability from the data set to leave us with just the remaining biological signal that we are presumably more interested in. Different protocols differ in how they process the RNA, amplify it, and so on, and things like that. And the different protocols might differ in terms of their coverage of each transcript, bias towards AT or GC content, their ability to capture short transcripts and things like this. And these potentially also introduce other biases or other batch effects that could differ between times when we've processed our cells, or otherwise just introduce variability that we're not specifically interested in, just comes along as nuisance and that we might want to account for. So with that, time to try this for yourselves. So using the read data, the aim is to repeat this analysis and see what it looks like for the read data, and then compare that to what we saw with the UMI data, and also compare that to the solutions we came up with.